Hey everybody, welcome to another lecture video. Alright, so chapter 21. We're now going to look at how those proteins that we looked at in the last chapter get synthesized and how DNA actually encodes that information for the primary sequence of those proteins, the order of the amino acids. So we're going to have to look a little bit at DNA and how DNA is organized, the way DNA encodes that information, and how proteins actually come from the DNA. Histology technician. We can learn a lot by studying proteins and different versions of enzymes. I think we may have looked at isoenzymes, remember, that can be used as diagnostic tools. So I guess a histology technician is somebody who can actually identify diseases or diagnose a disease by the presence of, or absence of in some cases maybe even, of certain molecules in our bodies. It's neat. Alright, so nucleic acids are the way that our body stores that genetic information. And so the nucleic acids, there's 4 of them in DNA and 4 different ones in RNA, for a total of 5 common, and then there are some uncommons. So yeah, if you thought I had bad math there, some of the nucleotides are going to be reused in DNA and in RNA. So a nucleotide is what we're looking at here, and a nucleotide consists of a nitrogenous base. This is going to be usually a purine or a pyrimidine, a sugar, and these are going to be deoxy or, well, I guess I should just say they're going to be ribose or deoxyribose. This is a pentose sugar, and a phosphate group. So all 3 of these things together is a nucleotide. Now, a nucleoside is just these 2 things. And sometimes you'll hear that term before nucleotides get made. Of course, they need to be nucleotides. I'm sorry, nucleotides have to come from nucleosides. It is worth noting that DNA is made from RNA, and I don't mean synthesized into like RNA is copied into DNA. I mean that in order to make a DNA nucleotide, you have to make an RNA nucleotide and then remove an oxygen. So deoxyribo nucleic acid and ribo nucleic acid. Ribo comes from the sugar, which is a ribose. Okay, that's enough about nucleotide. So there are different kinds of nucleic acids. There's DNA and there's RNA, and I just mentioned that the main difference is going to be that structure of the sugar. DNA is what stores the information. RNA is what we're going to see is used as an intermediate. So we'll copy our DNA into RNA, which can then be turned into proteins. Nucleic acids are large molecules. I think what it's referring to here is the overall finished, structure, not the individual nucleic acid, the nucleotide. But our DNA or our chromosomes, these take up a lot of space in the cell. They're found in the nucleus of eukaryotic organisms like plants and algae and humans, right? Spiders. But in prokaryotes, there is no such section. Everything just kind of floats around within the cell. There's no nucleus. But they also have nucleic acids. We don't want to forget that. And then it stores the information, and in some cases can direct big, what is the word I'm looking for? They can direct cell responses. So in some cases, just growing cycles or mitosis, when cells split in half, can be directed by movement or reorganization of nucleic acids. In our example of our bodies, there are times when our cells want to duplicate. So for that to happen, first our DNA copies itself. And once there's 2 copies of that DNA, then they migrate to opposite ends of the cell, and that's sort of, right before the cell's going to ultimately break in half. And all of this stuff, all of those actions are sort of orchestrated by what the DNA is doing. So here are our bases, our nitrogenous bases. We're going to have either pyrimidines or purines, and if you remember back to our amines and amides chapter, there was those nitrogen-containing rings, right? We looked at 5-membered rings and 6-membered rings, and of the 6-membered rings, these are 2 of them, purines and pyrimidines. Now they, depending on which, whether it's a purine or a pyrimidine, and then depending on subtle variations to these structures, will determine which nucleotide we're going to be looking at. It was also mentioned up here that there's hydrogen bonding that's available or that's capable between these guys. And so we're going to see that hydrogen bonding is actually pretty important with holding our DNA together, but also in how our DNA kind of winds into a double helix. Okay, so here is the, these are the 5 bases. These are the 5 common bases. There's some uncommon bases, like one designated I, that shows up in tRNAs. So these aren't the only ones. Like even with amino acids, right? There's 20 common amino acids. There's one considered the 21st amino acid, selenocysteine, and then there's even methylated versions like methylalanine and hydroxyproline. So there's a lot of wiggle room when we talk about the number of things. But in this case, there are 5 common bases. There are 4 that are shown or that are found only in DNA. That's thymine, cytosine, adenine, and guanine. And I shouldn't say they're only found in DNA, because obviously some of them are found in RNA. What I mean is, in DNA, we only see these 4. We see C, T, A, and G. So the 2 pyrimidines are cytosine and thymine, and the 2 purines are adenine and guanine. And that's always true. We see 2 of each. And so in DNA, that's what we see. In RNA, instead of thymine, we see uracil. And so that's the difference. And so we still see these 2 purines in RNA and in DNA, but pairing with, or not pairing with, but accompanying cytosine as the pyrimidine representatives in RNA and DNA, uracil is in RNA and thymine is in DNA. Okay. So you can see there are structural differences there. And again, this is the same slide. It's just highlighting again the 4 different RNAs, nucleotides that we see. And again, these are the nitrogenous bases. There's also a difference in the sugar, and we're going to see that here in a second. So as far as the sugar goes, you can see on the top here, we've got ribose, and look at the number 2 carbon in our ribose sugar, and you see an OH group down below. In the deoxyribose version, so down here, we see that OH is now an H. So that's the difference, and so nature makes this molecule first, and then an enzyme will remove that oxygen. And so that's why RNA is the precursor molecule to DNA. Literally, to make DNA, we need to have already made an RNA. Nucleosides are that first reaction, that initial nitrogenous base combining with the sugar. And we call that a beta-N glycosidic bond. We know that when sugars form, essentially when those hemiacetal sugars react, they form a glycosidic bond, so that's where this is coming from. We're calling it an N glycosidic bond because it's attaching to an N of the nitrogenous base. And it's beta because the OH is up. So beta-N glycosidic bond. Now depending on which nitrogenous base it is, that's what we're going to name it as. So when it's an adenine being added, we call it adenosine. When it's a let's say cytosine. I'm sorry, what is that? It's not called a cysteine. Sorry, I always get tongue-tied after I do an amino acid. Cytosine, yeah, cytosine. When it's a cytosine, we would call it, well, what would we call it? Would we still call it cytosine? Here we go. No, we wouldn't. We would call it cytidine. So, yeah. So the naming is a little funny. So adenine becomes adenosine, guanine becomes guanosine. Cytosine, seems like they should have named it cytidine so that they could call it cytosine and fit with the trend. So cytosine is called cytidine, and then uracil becomes uridine, and thymine becomes thymididine, or in this case deoxythymididine, since that's in DNA only. So those two names are here, sorry. So the deoxyadenosine, deoxyguanosine, they match the normal other name. Deoxycytidine, uridine only in RNA, so it doesn't have a DNA name. All right, so I skipped a little bit. Let me jump back. So that was our nucleosides. And then the nucleotides, the phosphate group is added. We call this a phosphodiester bond. When we start to link nucleotides together, we're going to say that there's a phosphodiester bond, because there'll be an ester from the phosphate group to one sugar, and then there will be an ester from another phosphate portion of the phosphate group to another sugar. Okay, so our phosphate, sugar, and base all together makes the nucleotide. Here's worth noting too that when we talk about DNA, we're going to say that it's read from 5' to 3'. What it refers to when we say that is actually this 5' sugar that we're seeing right here, this 5' carbon on the sugar. The 3' is going to be this carbon right here in the same sugar. So when the next chain grows in, or the next phosphate group comes in, it's going to be reacting with, the next sugar and so on. I ran out of room to draw my sugar. There's going to be a reaction here from the 5' of this sugar to the 3' of this sugar, and we're going to say that there's always a 5' side and a 3' side, a 3' hydroxyl. Okay, just throwing that in there because it's coming. So we looked at our nucleotides, and then here we get kind of we don't get any kind of naming help here, but okay, basic ingredients. Here we get that naming guide. So if the purine, I guess, ends in ocene, it gets its name changed to idine. And we add deoxy in front when we're talking about DNA. We would also say monophosphate. I was neglecting to talk about that part of it. We can talk about that part of it. So some of you recognize AMP as being what happens when ATP is completely hydrolyzed, right? When we hydrolyze ATP into ADP, a phosphate comes off, or we stick it on somewhere, or it gets transferred, but it liberates energy, and ADP ultimately can go to, I forgot what I was doing, AMP, adenosine, or guanosine, monophosphate. And so this is also a backwards direction. We can make these high energy molecules like ATP from AMP. So these are the same molecules. These nucleotides are the ones that are involved in those high energy reactions. But when it only has that one phosphate on it, it's called monophosphate. And then here we get that full list of names. So you see the bases of DNA and RNA, the nucleoside names of DNA and RNA, and then the nucleotide names, and also their sort of designations. jumped the gun a little bit with my connection to ATP, but here you can see that reaction. So when you add the phosphate group, AMP grows to be ADP, adenosine diphosphate, and then ATP. So you can, you don't have to memorize these. Obviously, the testing situation is quite going to be different this semester. So in one world, you might have had to be able to look at that and know that this is a purine, and then know if this was adenine or guanine, but not now. You should be able, though, I guess I don't have to go all the way back. You should be able to look at this, though, and know if this is RNA or DNA. There's your clue right there. All right, so now let's talk about DNA itself. So primary structure, similar to primary structure in proteins, is going to be the sequence of the DNA. And the way that the different nucleotides are linked together is through a sugar phosphate backbone. There's a phosphodiester bond. We've talked about that one before. And so here you can see the 5' end of this sugar, the 3' end of this sugar, and the 3', or well, that's not the 3' end. This is the 3' end down here. And we've already seen that there's a bond that's taken place. We've got a phosphodiester bond. Okay, so the way that it grows, remember, we've got the 3' end and the 5' side of the incoming nucleotide. So we're going to grow from 5 to 3. Sometimes there's a lot of confusion around the directionality of certain strands and which ones can go what direction. And we always will read DNA 5 to 3, just like we always read proteins from N to C. Okay, so primary structure is just the sequence of them, the way they're all joined together. We can see a schematic here of the backbone, general backbone. It's going to be phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. The nucleotides themselves are connected to the different sugars, and so they're not part of the backbone. So this is the primary structure. The 5 to 3 direction carries the genetic information. Whenever we write that, one of the simplified ways of writing the sequence is to show it like this. There is some more complicated ways that add little p's and whatnot, and so on, depending on which side has the phosphate, and we're not going to get into any of that. 5 side, 3 side. RNAs, very, very similar. The only difference is instead of having thymine, we have uracil. Same direction, same way we read it, 5' to 3'. Now, before we talk about how DNA is turned into proteins, we'll look at how DNA gets replicated. And so first, DNA is associated with another strand called the complementary strand in a double helix. So we'll investigate a little bit about how that double helix is held together. And so there are base pairs that we'll talk about. We'll talk about adenine pairing with thymine. We'll talk about guanine pairing with cytosine. And the reason that they pair is because they have the ability to hydrogen bond to each other. Now, because adenines pair with thymines in the DNA and in its complementary strand, there's equal numbers of them. And so right off the bat, if I was to say that there is 22% adenine, you'd be able to figure out how much DNA is, or how much of the other base pairs there are altogether, because out of 100%, adenine and thymine are going to be the same. So adenine's 22%, so thymine must be 22%. That's 44%, right? Well, out of 100, what percentage does that leave for guanine and cytosine? I think it's, what, 56? So if it's 56%, then what's half of 56? Because that's what the guanine will be. And so you can see how this is done. What is it, like 28? Or something like that. No, that's not right. Yeah, that's right. So knowing one of the nucleotide percentages, you could find them all out. But the base line that I'm trying to set or illustrate here is that they're 1-to-1 ratios with each other. So however much adenine, that's how much thymine there is. Let's look at how, well, we'll look at the base pairing in just a second. One of the ways that scientists used to compare organisms long before we could sequence the actual DNA and look at it that way, which is by far more meaningful, they could look at just the percentage of how much A and T, G and C you had. There's also something called the melting temperature. Different organisms will have different ratios of A, T, and CG. There's actually 3 hydrogen bonds that take place between C and G and only 2 between A and T. And so organisms that had a lot more GC, their DNA didn't separate as early, or as it took higher temperatures to get it to separate because it had a lot more of the stronger bonds or just additional bonds. But again, what my point is, before we could sequence, we were just looking at how much or what percentage of CG or AT an organism had and then comparing them. But you can see here that doesn't really tell us much. For example, we have, chicken and salmon have roughly or exactly the same percentage of base pairs, but chicken and salmon are obviously very, very different. I think humans and E. coli, which isn't on this list, also are very, very matched, or humans and yeast or something like that. But of course, we're completely different. So you can't make any meaningful comparisons based on this. This is just a number that we can look at, but it doesn't end up meaning anything. The base pairing, let's take a look at that. So complementary basing takes place between 2 strands of DNA. Again, we're going to have one strand of DNA running this way with all its nucleotides, and then the other strand will be running in the opposite direction, 5' to 3' as well. And the base pairing takes place between the chains. Adenine and thymine have 2 hydrogen bonds that they can do. Guanine, cytosine, 3 hydrogen bonds. So again, this is why it takes a hotter temperature to get DNA that's really, really rich in CG to separate, and it's really easier to get AT-rich DNA to separate. When we get to the actual transcription, which is transcription or replication. Transcription, I think, though, specifically, where certain regions where the DNA actually has to separate so that your protein machinery can go in there and copy it and start, reading it. Those parts of the DNA that are supposed to separate in order for those proteins to get in there, those are going to be really high in the ATs and have not so many GCs in there because ATs are easier to pull apart. So your body takes advantage of this difference as well. So the DNA winds into a double helix. So the 2 strands sort of curl around each other. The base pairs, sort of the way they align, is kind of like if this is, and then we get sort of stacking. I lost my picture here. I can't draw it anymore. But the base pairs sort of wind around each other, and then because they're flat, because these ring structures are flat, they sort of stack really nicely. So you should pause the video, give this a shot. You're going to write the complementary strand A pairing with T, G pairing with C. And see if you can come up with the right sequence. All right. So hopefully you came up with that sequence. A lot of times students are confused about the direction that DNA is written. DNA is always written into the 5', 3' direction, and that just means the machinery that we have that copies DNA writes it that way. So you might be wondering, well, then which strand does it read when it's copying, right? Well, let's talk about that real quick. So the 5' to 3' strand, let's call that strand number 1. This encodes the protein. This encodes the amino acids. These are ultimately going to be turned into amino acids if this is a part of a gene. Well, if we were to copy that, we would get this sequence down here, which is not the same. And then you say, well, you don't read it 3 to 5, you read it backwards, right? You read it this way. But AGT you don't find in this backwards direction. There is no AGT. So that information is in the one strand, but not in the complementary strand. So when DNA is copied, and when DNA is turned into mRNA for translation into proteins, it's not the number 1 strand here that gets the coding strand that gets copied. It's actually this one here called the template strand. And so the 3 to 5 strand becomes the template strand, because let's say your machinery comes in here to then make a copy of the DNA. If it comes in here and it pairs up in the 5' to 3' direction, an A, a G, a T, a C, an A, an A, a T, a C, and copies the template strand, we get our coding strand again. We get that information transferred to a new molecule. And so that's where sometimes the confusion comes in, is students are trying to understand that the DNA is written from 5 to 3, and so when they get a piece of DNA like this, like the number 1 strand, they look at this end and then try to write a strand from 5 to 3, and you end up with a sequence that's different than the one that you would get. Well, I guess that's not true. Whatever you would draw would ultimately be this thing, but that's not the strand that we want. The 5 to 3 strand that you just drew is not the strand that you would go and interpret to try to get protein information from. So again, don't worry about that right now. But that's just a lot of where the confusion comes in, is just when we're talking about the direction that it's read or that it's written. So I thought I would just talk to you a little bit about where the information actually is, and then when we're copying the DNA, we're actually going to copy that second strand to make sure that we still keep that information we want. So DNA replication first takes place in our cells right before they're going to replicate, or to divide. And the cells want to make sure that each cell, each new daughter cell, has all of the genetic information. So before copying can happen, DNA has to first separate. The 2 strands have to separate. And what we're talking about here is the simplest case, all right? This is probably in prokaryotes. I can't spell. Prokaryotes they don't have nucleuses, they don't have a lot of complicated machinery. When we go to eukaryotes, everything changes. We have a nucleus, so all of the genetic information, all the DNA is stored in that membrane-bound or membrane-enclosed sac inside the cell. We have DNA that's associated with proteins called histones. They're wrapped around these histones. Our DNA is also coiled into something called chromatin, which condenses into the chromosomes. So when we talk about DNA replication, and I show you these next couple of slides, realize that this is in the simplest light. This is probably and even when we study it in eukaryotes, it's yeast that we study it in. So this is just bacteria, which is not as complicated. But with bacteria, even before things can get started, the two strands of DNA have to separate from each other. We'll call the original strand the parent DNA, and the two new strands the daughter DNAs. So you can see the replication fork. Now, the replication fork is where the DNA is unwinding. Now, the direction of replication at the moment is taking place in this direction. So there's already something called the replication bubble, and if you can imagine, you've got your two strands of DNA, and then there's this bubble, and in that bubble there's new strands being formed, and then there's the replication fork right there, and that's where these two strands are going to open up and allow more replication. So what you would see in a couple of seconds, I suppose, would be an even bigger bubble with all these new daughter strands growing like that. But anyways, that's what we're seeing in this other picture here. And so the template strand here is being read by something called DNA polymerase. DNA polymerase is sitting on this, riding along this template strand, building this daughter strand. And the daughter strand is in blue down here and in the 5-3 direction. And so these are the, over here, the new little bits being added on. Now, what happens with the other side is a little more complicated because the DNA that's being copied on the other side needs to be read in the other direction, right? Because let me kind of explain. The 5' side is here, but the template needs to be read from 3 to 5 so that the DNA can be built from 5 to 3. So that means DNA polymerase, which there's only one of, and DNA polymerase is, you know, kind of spanning this whole area here, has to read the purple strand in this direction so that it can build the DNA in the correct direction, the 5' to 3' direction, which is this direction, right? The same direction that it needs to read this other strand. So there's some neat little folding around that happens. The book doesn't have a good picture of it. I'll post a link to a really neat video that shows it in action. It's really cool. It's a CG video. But because of that, there's two strands. There's the daughter strand that's continuous, called the leading strand. And it's formed continuously because it's just made as DNA polymerase moves along and you know, approaches this replication fork that's constantly unwinding the DNA. The other side, though, is called the lagging strand because it's actually synthesized in little chunks that have to be pieced together later, and those little chunks are named after the person who discovered them. They're called Okazaki fragments. So before the replication fork happens, there needs to be unwinding of the DNA, the double helix, and that's done by something called helicase. It'll unwind the DNA and start to pull the strands apart. Once those strands are pulled apart, there's these little binding proteins that come in and sort of protect those now exposed nucleotides from side reactions while waiting for DNA polymerase to come along. We'll talk about DNA polymerase in a second. Some of the requirements in order to get DNA replication happening is you have to have plenty of those nucleotides. Well, they're not nucleotides yet, right? They're nucleosides that are going to be reacted with that growing chain to then become a nucleotide. You got to have that phosphate backbone to be a nucleotide. Okay, so as the template strand is being read, every time DNA polymerase encounters a T, it's going to stick in a new A into that growing strand. It's going to be putting whatever would base pair with it, whatever would have normally complemented that nucleotide. So at the end of replication, you're going to get a new strand of DNA that looks or a new double helix that looks exactly like the parent double helix. Now, when we say exactly like, there is mutations that happen. We'll talk about all the errors that happen and also the proofreading that takes place. But still, mutations do happen, and so ideally it would be exact, and it probably is the majority of the time. The way that the proofreading mechanism sort of works is just by checking to make sure that the base pairing of the new strand or of the new nucleotide that was put in is proper. Incorrect base pairing might lead to bulges or weird misfolding of the nucleotides that the body can detect and then try to fix. And yeah, so we'll talk about these Okazaki fragments in a little more detail. Oops. Okay, so helicase unwinds the DNA. DNA polymerase is the enzyme that actually is responsible for making the reaction of those growing nucleotide chain, you know, the daughter strand, the growing daughter strand. It moves in the 3-5 direction, catalyzing the new linkage, the lagging strand, also growing in the 5-3 direction, called Okazaki fragments. They get joined together by DNA ligase. That is the enzyme that joins the lagging strands together. So this again isn't a great picture because it doesn't show you how the synthesis of the lagging strand takes place. And it has to do with, yeah, I wish I could show it. If you can imagine taking this strand of DNA here and turning it over that way, bending it, so that instead of making that shape, it was like this shape, okay? Then you would be able to see that the DNA would be the purple one on top and the blue one on bottom, which is exactly like this strand looks like. And so it folds that bottom strand over so that they're both, so that it's pointing in the exact same direction as that other strand's pointing, this one and this one, and then DNA polymerase catalyzes the growing chains at the same time. Now, it can't hold this loop forever, and so then it lets it go, and then it loops a different part. And that's why we end up with little fragments. And then, you know, DNA ligase comes in and pieces those together. Now, for this also to work, it's not mentioned here, but we need a primer. We need an RNA primer. DNA polymerase doesn't know where to go and sort of start or where to sort of hang on. And so there's a little, usually there's a little, and you can see it here actually, this little red bit. This is a primer. This is telling, well, this is actually, this lands here through complementary base pairing, and it allows the DNA polymerase to actually make a reaction to something. There needs to be a nucleotide for DNA polymerase to stick a new nucleotide on. It can't just like start one spontaneously. So these little primers, you see a lot of them on the lagging strand. You don't see any primers on the leading strand because it would have been way up here at the very beginning. Okay, sorry about all the mess in here. See if there's anything else to mention in here. Helicase does the unwinding. There we go. Single strand binding proteins, these little yellow guys, they're the ones that are protecting the exposed base pairs. Of course, you don't see them over here. And then something actually has to come in and cut out the primer and then piece back, fill it with DNA, because that primer is made of RNA, and that means it's the wrong sugar. There's a few different DNA polymerases. There's actually a lot in our bodies. There's a few in prokaryotes. The main one that we're talking about here, I believe, is DNA polymerase 3, unless it's DNA polymerase 2. Doesn't matter here. They all do similar jobs. The main difference is that some of them can put DNA nucleotides in only the 5 to 3 direction, whereas some can put it in the 3 to 5 direction. Mainly that means when they've stuck in a nucleotide and they detect that it's wrong, they can actually cut it out, kind of like backing up a step, cutting it out and then putting in a new one. And not all the polymerases can do that, but that's part of the checking mechanism. All right. So pause that, give yourself a little quick quiz, and then here's the answer. Okay, so let's talk about making proteins. So that's replication. This is transcription. So transcription is the process of not copying the DNA for storage purposes, but copying it into a form that can be then turned into a protein. The main player in this sort of process is going to be something called the ribosome. In our bodies, we have ribosomes that are membrane bound to our endoplasmic reticulum. So we have a rough ER and a smooth ER. It's rough because it has ribosomes on it. And then we also have ribosomes floating around in our cytosol. We do not have ribosomes in our nucleus. Prokaryotes, on the other hand, don't have a nucleus, and so their ribosomes are floating around everywhere. And the difference is that as DNA is being, anytime DNA is floating around, ribosomes might be doing a job and making proteins. In our bodies, we regulate the process. So we make RNA, and then we make messenger RNA, and then we pump it out of the nucleus to let the ribosomes get at it. But these ribosomes are the thing that turns mRNA, which is essentially the gene information, into proteins. So RNA and DNA, we've already seen some of those differences. RNA makes up most of the nucleic acids in the cell. It transmits genetic information from DNA, as I mentioned, into those messenger RNAs. There are a few different types of RNAs as well. I think we're going to go over them. There's tRNAs, there's messenger RNAs, and then there was the ribosome, which I probably could have mentioned was an RNA. It's actually a mix of RNA and protein. I forget what the exact term for what that hybrid is called, but a lot of the mass of that RNA or that ribosome, is protein, but it does have RNA catalytic activity, meaning that the parts of the ribosome that are catalyzing the reaction is actually the RNA portion of it. Okay, let's see, we know the difference between the sugars. We know that uracil is there instead of thymine. Here's another difference we didn't know. RNA strands are single, and DNA is the double helix. So we don't see RNA forming those double helixes, though we do see RNA forming secondary structures, secondary and tertiary structures, which we don't see DNA doing. And then the molecules themselves are much smaller than DNA. DNA, of course, has all the information in it. RNA molecules tend to either be functional, like an enzyme, like a ribosome, which is a small portion, a small molecule compared to the big genome, or they're messenger RNAs, which are only one gene long. Again, and DNA has all the genes, so that's what they're basically saying here, is that there's no real comparison in size. 5% of the total RNA that we find in the cell is the messenger RNA. Once it gets made, it quickly gets broken down, and that makes sense once you think a messenger RNA gets turned into protein. So, unless you're trying to make that protein forever and ever, you want to regulate that. So it gets broken down pretty quickly. Transfer RNAs are also important. Transfer RNAs are the thing that is going to recognize each of our amino acids and actually have a role in building the protein. But 80% of the RNA, or at least by mass, is found in the ribosomes. And the ribosomes are very, very big. and they are the site of protein synthesis. Typical RNA molecule, we're going to look at a picture. In fact, let's just look at the picture here. So these are the two ways that it's shown. This is the cloverleaf form, this is the L form, for obvious reasons. This is what I meant by secondary structures. This RNA molecule is hydrogen bonding with itself in a few places, right, to form these weird little structures. Now there's an acceptor end, this 3' end. This is going to actually recognize the amino acid. So this is particular. Every amino acid we have has a tRNA specific for that amino acid. And some amino acids even have multiple tRNAs that will recognize it. And so the tRNA will recognize an amino acid. Let me just draw an amino acid and say that it's going to stick right here at this end. Now, the mRNA, the messenger RNA that has the information such as which amino acid goes where and in what order, that's going to communicate to the tRNA via something called the anticodon, this region down here in red. Now, there's also something else called the Trna Synthetase, which is responsible for actually attaching the amino acid to the tRNA, and those can recognize different parts of the tRNAs. Some of those recognize this little region over here, some recognize the acceptor end, some actually look at this end, some look at little spots on the inside. So that's how specific some of these enzymes that do the attaching of RNAs to amino acids can be. Okay, so that's the tRNA structure. So let's look at mRNA. So mRNA is really the process that we're going to focus on the most. It's similar to DNA in the fact that DNA has to be unwound, and then an enzyme comes in and copies it. Now, where we had DNA polymerase do it the first time, here we're going to have RNA polymerase. And RNA polymerase's job is to make an RNA strand from the DNA. And so the general transcription process takes place in the nucleus. The DNA is going to produce the mRNA strand. Now, in our bodies, we have something called pre-mRNA because we have introns. These are I want to say they're non-coding portions of our DNA. They don't code for a gene, but they're, speckled throughout our genes, and so those have to get cut out. But once we have a processed mRNA, that gets put out into the cytosol where ribosomes are, and ribosomes will do the second half of protein synthesis called translation, and we'll focus on translation after we look at transcription. So transcription takes place in the nucleus. We're going to make that mRNA. So the only difference really from the replication sort of machinery is that new strand, the daughter strand, isn't going to have any thymines in it. It'll have uracils. The direction is also the same. We're going to read the 3' to 5' strand so that we make a copy of the 5' strand. And so mRNA is going to be made in a 5' to 3' direction. And you can see it sort of zoomed in here. We've got the growing strand is just incorporating these new nucleotides. Now, there's 3 steps to this. There's initiation, there's chain elongation, and there's termination. I don't believe our book is going to go into any of these details. Let me see real quick. Yeah, not really. Okay. So I'll just briefly say it. So initiation is just, how does the RNA polymerase know where on this DNA to go? Well, there's usually specific sequences that it'll recognize called promoters. And DNA or RNA polymerase will go and it'll find those. A few other proteins come in called transcription factors and initiation factors. And when all of the machinery is there and all of the players are there, then it takes place. Chain elongation is just what's happening here. We're seeing this moving along the DNA, copying the mRNA. Now, termination happens when it hits something called the termination site. Now, a termination site could be it reading a particular sequence, and that's usually what happens. They get to a certain, or they code a certain sequence that gets put into the mRNA that causes the mRNA to make these things called hairpin loops. It causes it to fold on itself, similar to that cloverleaf structure we saw with tRNA, which helps cause or tells the ribosome it's time to stop, or time to come out. Sometimes it also causes just stalling, and then there's another little protein called Rho, and the Rho protein goes and chases down, finds the mRNA, and then it just starts chasing the ribosome, and it'll catch the ribosome when the ribosome stalls out on those like hairpin loop structures, and then when Rho gets over here, it dissociates everything. So a little more detail than you needed, but that's kind of how it goes. Now, I mentioned we have introns, which are these non-coding regions, and exons are the coding regions, and so those get cut out. They get processed by something called small nuclear RNAs, or SNRPs, and then there's also some other kind of RNAs, little RNAs that do these jobs, microRNAs, and there's interfering RNAs which do different jobs, but there's nuclear RNAs that cut out these introns and sort of process the mRNA. So from that pre-mRNA to mRNA, and then that gets pumped out of the nucleus. Okay, let's see. So transcription is regulated a few different ways. One of those ways is by activators, which will actually go and bind to those promoter sites of the DNA. Let's say that your body's in desperate need of some new proteins. Let's say that you're deficient on some proteins, so you got to turn on that gene. And so usually there's an activator, some kind of molecule that's only present when you're low on those proteins, and it'll go and it'll bind to a certain region on the DNA right in front of the gene that you want to turn into mRNA, and that little activator will kind of recruit Rna polymerase where Rna polymerase may not have normally gone there to make that gene or to transcribe that gene. And so there are activators, and then there are also inhibitors that would go and sort of silence those genes. And so one way of regulating these processes is through activation or inhibition, in the means of proteins going and binding or not binding to those specific sites. Let's see. They usually call all of the area upstream from the gene the operon, but again, this is more information than you probably need here. Okay, so the proteins, the transcription factors often interact with that Rna polymerase. And you can see that here. Promoter region, transcription factor. I like this little, they're showing us here too. If this transcription factor in blue is required for polymerase to come and land here and start making this gene, you can imagine each gene would have its own transcription factor that would get polymerase right in front of that particular gene. Then this little guy here is an allosteric inhibitor, which comes in and, obviously when this one is bound, when these guys are associated together, polymerase is around. But if this guy wasn't here, so if something else was there, like an inhibitor that also bound there, then this blue little guy may not have been there. So different interactions with the proteins can lead to turning this process on or off. And so you can write the sequence of mRNA that would go here. Remember which base pairs are in mRNA versus DNA. Give that a shot. All right, so let's talk about protein synthesis, translation basically from the mRNA into actual protein. And what you're looking at here is, of course, that tRNA molecule that we saw a little earlier. And in this case, it's already got a serine attached to it, so it's activated. And then down here we see the codon, which is going to recognize the genetic code and actually read the mRNA in terms of the proper sequence of amino acids. So let's take a look. So protein synthesis starts with transcription. We just looked at that. That's the process of making the mRNA. We saw that with with eukaryotes like us, there's introns that get removed, so pre-mRNA gets processed, and then it gets pumped out into the cytosol, the aqueous portion of the cell, to interact with ribosomes. And so that's where, on the ribosome, where we're going to get translation. Now, the genetic code refers to 3 nucleotides at a time, and we would read these from 5' to 3' on the mRNA, and we're going to call them codons. Now, there's codons for each of the 20 amino acids, and then there's our stop codons, and then there's a start codon. So let's take a look at those. Now, we haven't seen the code yet, but this is what we would basically do. If we had an mRNA section, we could take a look at each of those and break it into the codons, and then we would look at what CCU actually codes for. If we look at our chart, we'll see CCU codes for proline, AGC codes for serine, and so on, and then we would have our sequence of amino acids. So on the test, I'll probably give you a 5' to 3' mRNA and ask you to give me the protein sequence. So here's that genetic code. So you can see here all 20 amino acids. Some have 4 different codes that will code for the same amino acid. Others, there's some differences. We get pyrimidines if the third position is a U or a C. We get purines if it's an A or a G. There's a couple families that way. Sorry, that's not one. The ones where there's two different amino acids with the same 2 initial letters, the purine or pyrimidine actually makes a difference here. Anyways, different codes. Now for prokaryotes and eukaryotes, we have a start codon AUG. For most organisms, that codes for a methionine. In prokaryotes, it's a formulated methionine, called FMET. And then there's some stop codons. So there are no tRNAs that recognize these stop codons. Instead, there are proteins that actually are related to the termination of translation, and these proteins will associate with these stop codons. But for another way of thinking about it, if there aren't any tRNAs bringing amino acids in, when we reach these letters, these codes, well then the protein chain stops. You'll notice that for a lot of these amino acids that have 4 different codons, that it doesn't matter what the last codon is, what the last digit of the codon, the last nucleotide. And we call this the wobble position. And they think that in nature, this helped with copying errors, so that in case that third letter accidentally got swapped out or was mistakenly written as something else, that it wouldn't change the amino acid. And we'll talk about how mutations affect the protein in a few minutes. So there's our genetic code. So once the tRNA is activated, it has the correct amino acid bound to it, and then that tRNA has an anticodon which will come into the ribosome, and we'll see that here. Actually, we don't see it here. We see the top of the picture. Let's look at the bottom of the picture. Here we go. So here is the mRNA being translated. Now, somewhere over here at the beginning would have been our methionine, our start codon. But then every 3 digits is another amino acid, and so on. And we can kind of see the rest of that picture up here. And so the ribosome has a few positions. It's got the E spot, the P spot, and the A spot. Now, there's initially an amino acid, a tRNA will bring in an amino acid to the P position, the peptidyl site. And then this is the acylation site. So the incoming tRNA that's bringing the next amino acid comes into the A site. And so here you can see it's bringing isoleucine, and the P site already has this growing peptide chain. Well, what'll happen is, and then in the E site, that's where an empty tRNA leaves. So this tRNA that's leaving just brought in, or this tRNA that's leaving probably was holding the chain right here at the phenylalanine. And then the tRNA that brought in this alanine transferred the whole peptide chain over to it, and then this tRNA was now empty, so it leaves. Now the next chain's ready, or the next addition. So in the A site, we get this tRNA bringing in isoleucine. A reaction is going to happen, and this peptide is going to get connected to isoleucine, and then the ribosome scoots over. It'll scoot over one position, and actually what it does is it pulls the mRNA through, but for all intents and purposes, the codons shift over. And now the A site, where we had this amino acid, this tRNA, is now the P site. The A site is empty and waiting for the next tRNA, and now the tRNA that was here in the P site is in the E site, and it's going to leave. And so that's what happens. The ribosome just moves along the mRNA. The tRNAs come in and recognize the next anticodon, or the next codon with their anticodons. And so we can see here the next site will be a GUC. And so whichever tRNA has a CAG on the bottom of their tRNA, that will come in and recognize this next spot and bring in the next amino acid. And so this will happen until it reaches a stop codon, at which point it stops. And so this is the translation process. It requires a ribosome, which has a couple of subunits here, one in purple, one in blue, and a variety of proteins all associated together. So here's kind of showing that process. Here's the DNA, copies into the template strand, or the template strand is used to make the mRNA, which you'll notice is essentially the same as the DNA, except it has uracils instead of thymines. The tRNA anticodons are here, and then the amino acid that this encodes, or the amino acids that it encodes. So go ahead and give this one a shot. Pause the video, see if you can come up with the order of the 3 amino acids. Of course, they're giving you the codons here, but otherwise you would look at that table that had all 20 on it. Okay, give that a shot. Hopefully you came up with the correct sequence. So let's talk a little bit about mutations. Mutations are going to be changes to the protein sequence or changes to the DNA, either through mutations that happen to the DNA itself or errors that happen in the translating process. So a mutation, change to the DNA that may lead to changes in the structure and function of cells. These can be caused by mutagens or carcinogens, things that mutate, things that cause cancer, radiation, certain types of light, and certain chemicals. And there are some viruses also that can cause cancer. When a mutation is bad enough that it alters a protein or enzyme and that the person can die from it, that's what we would call a disease, and that would be a genetic disease because it's just built into the DNA. So a normal DNA sequence is shown here. Normal DNA sequence would normally give you a good mRNA, and a good mRNA will translate into a protein strand. So let's look at how a mutation affects that. So we'll compare things to the methionine, lysine, phenylalanine, glycine tetrapeptide. So that's normal. So what kind of mutations are there? Well, there's a point mutation. That means we just switch a letter. We take a C, for example, and turn it into a T. So where before this strand said CCG, we can go up and look at it. See? CCG. Now it's changed to TCG. Well, the T is going to have a different pair with the or be copied into an A on the mRNA rather than a G. And so then instead of having GGC on the mRNA, we have AGC, and that codes for a serine. Excuse me, a serine instead of a glycine. Now this is bad. Glycine is a small amino acid that doesn't have a charge. That's nonpolar. Serine is polar. So having a polar amino acid switch for a nonpolar one might lead to all kinds of problems, like proteins not being able to fold properly, tertiary structures that don't hold together. So that's a bad mutation. A silent mutation is one that doesn't change the amino acid. So for example, what if in the wiggle room, remember the wobble position, let's say that we changed one of those. Let's see, do I have an example? I don't have an example, but let's talk about it. In a silent mutation, let's say I changed, this AAA here to be like an AAT. Well, that would lead to a UUA instead of a UUU. Let's see if that makes a difference. UUA instead of UUU. We're going to jump all the way back up here. Let's clean this up. UUA instead of UUU. Okay. So here's UUA and here's UUU. So that actually would make a difference. We'd get a different amino acid. So maybe that was a bad example, but let's use this. CUU versus a CUA. Having a mutation that changes a letter to another letter that doesn't change the amino acid is what's called a silent mutation. It is a mutation, but it's silent because nothing happened in terms of the protein. The protein came out the same. Silent mutations are good. A deletion mutation or an insertion mutation. This adds in a new nucleotide. So there's one that was there that's gone, or now there's a new one. And both of these are called frame shift mutations because they change the reading frame. So let's take a look. If I come in here and I remove that A, remember there was 3 A's here, AAA. If I take it out, well, now the codon is AAC instead of AAA, and the next codon, which should have been CCG, is now CGA. I've shifted everything over. And so now I'm going to get all new amino acids. The AAA is now going to give me leucine. The CGA is now going to give me alanine instead of glycine. So this changes the whole sequence of everything. So of the worst kind of mutations, deletions and insertions. All right. So as we mentioned, some mutations can be silent. They don't cause significant changes. Drastic changes could be when the amino acids go from being polar to nonpolar, acidic to basic. They can result in misfolding. They can result in loss of activity. If these amino acids were crucial to the active site, then we don't have any activity anymore. Okay, and that could lead to other things, right? If an enzyme stops working because of this, then whatever its job was, whether that's getting rid of toxins or catalyzing a reaction, those things stop, and so that could lead to disease. So genetic diseases are just those that have that are caused by mutations to your code, your genetic code. And as an example, there's an enzyme that converts tyrosine to melanin. If that enzyme's defective, then you don't have any melanin, and this is the basis of albinism, not having any pigment in your skin or hair. So just a few genetic diseases here. You could pause the video and take a look at some of these. Sickle-Cell Anemia is actually caused by just a change of one amino acid. So again, a little self-check here. Pause the screen, test these out, and I'll show you the answer in 3, 2, 1. All right. Hopefully you got those right. Recombinant DNA is something we can do. We can actually take genes out of one organism and stick them into another organism. And we do this all the time with things like insulin. We used to harvest insulin from pancreases, I believe, of dogs and pigs. Now we just make bacteria produce the molecule for us. Bacteria aren't doing anything else, right? They're not busy. So we take the gene for insulin and we insert it into their bodies and let them pump it out. The way that we do that requires cutting the gene out of one organism, and for that we use something called restriction enzymes. We can actually tell it where to cut, program these things, and then they go. We've actually copied a lot of restriction enzymes from viruses, because viruses use this. They have ways of going and inserting their DNA into the existing host's DNA, and that's exactly what we're trying to do. So these restriction enzymes often come from viruses. And then we need to find a way to incorporate that new gene back into the new organism. This means pasting it back in with ligase into the genetic information within that organism. And then when that organism goes and starts to transcribe its genes, it'll transcribe the gene that we inserted. Now here's one way we do it. DNA of microorganisms like bacteria, they often have something called plasmids. They're little circular bits of DNA that they can actually share with each other. And so what we can do is we can take these plasmids, we can isolate them, we can cut out little chunks of them with those restriction enzymes. Then we go and we use a different restriction enzyme or the same restriction enzyme and cut out the gene that we want to insert into the bacteria. We mix this stuff together, we put ligase in there to join everything, seal the ends, and then we have these plasmids that have the gene of interest in them. And often when scientists are studying whether or not we can actually get the gene into the organism, they put extra genes in there. So for example, you could take a petri dish and put antibiotics in it, and then you can insert a gene, let's say, that has one part of it that codes for, let's say, insulin, and then another gene on here that gives you antibiotic resistance. So that only the organisms that actually get this gene and start to make the insulin will also be making the antibiotic resistance, and they're the ones that'll live on this plate, this agar plate. All the other organisms that it didn't work in, the unsuccessfully transferred DNA, they'll die because they're not making the antibiotic resistance. So it's a way of isolating and singling out. You do this to a bunch of bacteria, and then you just go and wait for bacteria to grow on the plate. And only the ones that are growing have the insulin gene because they also have the antibiotic resistance gene. So some of the other products that we get from recombinant DNA, in addition to insulin, you can look at those here. Polymerase chain reaction actually has a lot of uses. If we're interested in trying to see if you contain a particular gene, and we know the gene region that we're looking for, we can try to use PCR. This utilizes different temperatures to have DNA separate from the double helix, to get copied, so you have to provide nucleotides to come in here and add to both of those now separated DNA. And so we end up doubling the DNA, and this happens by increasing the heat, lowering the heat, increasing the heat, lowering the heat, and repeating these cycles over and over and over. DNA separates, new strands form, and then DNA separates, and those new strands all separate, and then they all form new strands of their own. Now, to amplify a particular region of DNA, we need something called primers. So let's just say this is a long stretch of DNA, but we're only interested in this particular section of the gene, then we need a primer that'll recognize the beginning of that gene, and then DNA will get added to that primer, and then we'll have our first, so this would have a long stretch on it, and this would have a long stretch on it. And then when that gets separated, and copied again, the primers again go and recognize the individual starting spots. By the end, we end up with only amplifications of that gene. And so it's a really neat process. We can use this as a diagnostic tool to see if you have genetic markers for, let's say, cancer or something like that, or if you have a diseased, a mutated gene that might give you a disease. We can also use this, as it's been used recently, to look for the coronavirus. So we sequenced the coronavirus's genome pretty early on this year, and so we were able to create primers that would look for its DNA in a sample of DNA. And so when they were doing these kind of DNA tests, these corona tests that take 2 or 3 days to get the results on, it was because they were doing PCR. They were taking DNA from a swab, and then they were amplifying it to make millions and millions of copies of it if that little section of DNA that they're looking for was present. And so it took a while to grow, to do the PCR to get large enough amounts of the DNA that could be detected. And if it's there, they'll end up with it. And if they're looking for particular parts of the coronavirus, then the fact that they were able to could make lots and lots of copies of it would be the proof that it's there. We use a different test now. It's an antibody test. It's a little faster to read. But anyways, that's how polymerase chain reaction works. Yeah, it can be used to see if you have particular genes. Now, DNA fingerprinting is also something else that we can do. We take an organism's DNA and then using a restriction enzyme that cuts certain portions of it, we can cut it all up and then separate it through a gel. Now, the pattern that it leaves is like a fingerprint. It's unique to that organism, and so then we can make comparisons that way, or use this fingerprinting to help connect a person to their individual DNA sample, or to say if organisms are related. Let's talk about viruses. Viruses are technically not alive. They don't self-replicate. They don't have all the machinery that you would need to be alive. They're just kind of like information. They're like a program that's laying there. They have DNA and it does something, but it's missing all the parts to be able to carry out those orders in the DNA. Our virus needs an organism to do all the work for it. Viruses can be RNA or DNA. Small amount of genes, somewhere between 3 and 200 genes. Doesn't have any of the parts, like I mentioned, polymerases and all that. It does have some protein, but usually this is just its outer coating and the mechanism it uses to inject its DNA. So sometimes like a syringe-like structure. Viruses have similar life cycles. They infect a cell, they put their DNA into the cell. Our machinery goes and copies that DNA and makes viral proteins, and then the virus assembles into new virus molecules and then either bursts out of your cell and goes and infects other cells, or there are types of viruses that actually will just incorporate their DNA into our genome, and then it just stays there dormant for a long time. So we can get infected when the enzyme of the virus pokes a hole in outside of our cell and gets inside. The virus can enter the cell and the nucleic acids will mix with ours. Proteases will go and process the proteins and produce a pure protein coat to make the new virus DNA. So the proteases, these things were encoded with the viral DNA. You don't just have proteases inside your body that make viruses. Those had to be produced. So the virus took advantage of your machinery to produce those for it. And then they get out of the cell. Lots of diseases are caused by viruses. You can see that list here. Retroviruses are neat. Retroviruses don't carry DNA. Instead, they have RNA. This is how we know that RNA can also sometimes be the genetic information that leads to DNA, and that it's not always DNA makes RNA, RNA makes proteins. So what the virus has, retrovirus, like the HIV virus, it has viral RNA and also something called reverse transcriptase. So it comes with its own protein. This protein encodes the viral RNA into viral DNA inside the cell. And then that viral DNA will then go and, replicate inside our cell. And then as it's DNA, we have all the machinery to go and interpret that into RNA and into protein. So the only difference, again, is that a retrovirus comes with its own RNA instead of DNA. So here's the HIV virus. HIV causes AIDS, acquired immunodeficiency syndrome. It hurts your immune system because it targets the cells that are, the main player in our immune system. If this thing targeted our skin cells, then we wouldn't call this, acquired immunodeficiency. It'd be like acquired skin cancer or whatever. So this HIV virus comes with an outside packing, an envelope that keeps it safe, and this allows it to live on some surfaces for some amount of time. Inside, you can see that it comes with its own reverse transcriptase. So it actually already has the protein it needs to turn its RNA into DNA, because you don't have that. You don't have reverse transcriptase in your body, so the virus has to come with its own. The sad part is that when this virus is replicating in your cell, it's telling your cell to make lots of reverse transcriptase that it can fill all its virus buddies with and then send them out. So your body does make it, but it doesn't come with their own instructions for it. The virus comes with those instructions. Retroviruses inject their viral RNA. The single-stranded DNA forms double-stranded DNA called a provirus, which gets incorporated into the host cell. So this happens as well. Not all viruses do this, incorporate their DNA into ours. It's a survival mechanism. That viral DNA now has a forever home inside your body, and every time your cells duplicate, they're going to copy that virus DNA also. This is why they say when you get, shingles or herpes, that doesn't go away, and that's true. And it can come back. It just lays dormant, and then stress and other things like that cause, the shingles to come back, or they cause the herpes to come back. Yeah. Okay. So how do we take care of viruses? So let me actually show this picture. I think the PowerPoint has a problem on it. So this is the virus cycle, right? It enters. Oops, I can't draw on this one. My bad. It enters. So one of the things we can do is try to block its entry. So we can, whatever molecule it recognizes on the cell, we can try to inhibit that. We can try to blow up their little molecule that's going to try to touch ours. We can try to go and inhibit that with inhibitors or things that'll bind to it. Try to block it. Okay, well, if it gets in, now what? Well, we can try to inhibit its ability to make DNA, to turn its RNA into DNA. And with that, we have these nucleotides that I'm going to show you. They're analogs. They look like nucleotides, but they're not nucleotides. And one of the main things they're lacking is the ability to grow into chains. And so if these things get incorporated into the viral DNA, the viral DNA stops. So that's another way to stop it. What about if we can't stop it there? Okay, so then it incorporates into our DNA, and now it's starting to get made, right? It's starting to get made. And the proteins, the viral proteins are getting made. Well, what we need to do is inhibit their enzymes. So protease inhibitors would be inhibiting the protease enzyme that goes and actually packages those viral proteins and makes new viruses. So then an inhibitor in that sense. So I mentioned the different nucleotides. Let's take a look at those nucleotide analogs or nucleoside analogs until they're phosphate bound. So these guys look like the nucleotides, but they're missing that reactive 3' hydroxyl. So these things will not grow into long nucleotide chains. All right, and we talked about these. Cancer is another type of uncontrolled replication, essentially. You have like 200 and something types of cells in your body, over 200 types of cells, right? Skin cells, eye cells, your taste buds, your cells in your esophagus, liver cells, heart cells, muscle cells. All of them are different. Every one of them is susceptible to cancer, which is it's just uncontrolled growth. The cells just start dividing and dividing and dividing, and this is what tumors are. They're just big, big collections of a cell that's growing out of control. Sometimes they're benign if they aren't anywhere, or they're kind of cell that can hurt you, and they're in places that can't hurt you. You get a big tumor in your earlobe, you're probably not going to die from that. If they're in other tissues and they start to interfere with regular body functions, well, then that's trouble. Cancers, again, we talked about, can be caused by chemicals or things that radiation in some kinds, sun radiation or natural radiation from the earth, like radioisotopes, and even viruses. There are some viruses that will go, and whatever they did to us, whatever they did when they stuck their DNA in us, turned our cells on in a way that they won't shut up, and they're going to keep replicating, and then they cause cancer, like the Epstein-Barr virus. So you can see here that we do have a lot of information about what kind of cancers can be caused by some substances. So we know asbestos is bad, and that causes lung cancer. We know that vinyl chloride is bad, and that's bad for our liver. And so here are a couple of those. Oncogenic viruses, so these are some of those viruses that cause disease. The previous slide was showing you some of the chemicals that cause disease. All right. Self-check. Go ahead and see if you can match these up properly. All right, and then I think we have just the end here. So We looked at nucleic acids. We looked at the differences between DNA and RNA in those nucleotides. We looked at the structures that DNA forms, the structures that RNA forms. In terms of mRNA, we know that that goes into protein synthesis. tRNA is required for amino acids, and that rRNA, ribosomes essentially, that's what that is. That's required for actually making proteins. All three are together, right? mRNA has the information in it, tRNA comes with the amino acids, and the ribosome is the site of translation. We know that codons can have mutations in them, and there could be point mutations, deletions, insertions, which are known as frame shift mutations, but also silent mutations. And then viruses. They can come with DNA or RNA, and if they come with RNA, they have to have reverse transcriptase, the ability to turn that RNA into DNA. All right, guys. Hope you're reading your book, and I'll be posting another chapter soon. Stay healthy out there. See you guys next time.